If you want to get going, I think it's good. Is everyone back? Probably no one's going to answer. Their mics are all off. All right, take it away.
Okay, thumbs up if you can hear me. Yeah, okay. My name is Jadiel Wasson, and I'm a postdoc with Susan Mango. You've just heard her introduce the concept of TADs and various regulatory factors. In our journal club, we'll focus on two of these, like I mentioned before, which are the enhancer and also transcriptional bursting. Today, I will introduce these topics along with various methods that will give you a background on these two papers, and they will help with our discussion that we will have tomorrow and Wednesday. So the topics I will cover today are enhancers, exactly what they are. Enhancers in the context of a model system. I will specifically focus on the two that they look at in the two papers, which include the Globin locus and also the Snell locus in the context of fly embryogenesis. And finally, I will discuss transcriptional bursting. Throughout this PowerPoint, I will introduce methods and tools that will help you understand the experiments that were performed in these papers. And the presence of the methods and tools will be denoted by the presence of this little guy in the corner. So what exactly is an enhancer? The technical definition of an enhancer includes that it is a short segment of DNA, anywhere from 50 to 1,500 base pairs. Enhancers also act as binding sites for transcription factors. And they can exist at various distances from their target promoter. So they can be proximal, meaning that they're close to their target promoter, or they can be distal, which means they can be very far away, on the order of 10 kilobases away. Furthermore, they should activate gene expression of their targets. And this activation is independent of their orientation in the genome. So how exactly do you figure out that a small piece of random DNA acts like an enhancer? Well, in the early 1980s, there were several groups of scientists that were in the business of trying to study gene regulation. One particular group was interested in the regulation of, say, this green gene. To study the regulation of this green gene, what they first wanted to do was introduce it into mammalian cells in order to get the expression of this green gene. To do this, they placed the green gene into two very similar vectors in order to introduce them into cells. However, by happen chance, one of these vectors included a small piece of viral DNA. So what do you think happened when they introduced these vectors into cells? Well, only the cell line that had the vector that contained that viral DNA expressed the green gene, which was quite unexpected. So they wanted to know, why do you only get expression of green gene in the presence of this viral DNA? They went on to figure out that this was a highly reproducible effect. And they revealed that the enhancing effect of this small piece of viral DNA was specific to a sequence. And it didn't matter the orientation of that sequence, how far away it was from its target, the green gene, or where it was relative to the location of the green gene. And as I mentioned before, all of these properties have now been generally accepted as the definition of an enhancer. Going forward, many groups in the field utilize similar setups to discover other potential enhancers that exist. In this assay that I'm showing you here, it's called an enhancer reporter assay. Here you have a minimal promoter that can't drive the expression of any gene without any help, attached to a reporter gene. And classically reported genes could either be LAC-Z, which when turned on will turn cells blue, or GFP, when activated, will turn cells green. So in this assay, what happens is, in order to determine whether or not a segment of DNA can act as an enhancer, they inserted it either downstream or upstream of this minimal promoter attached to a reporter gene in order to see whether or not this segment of DNA could drive the expression of the reporter. And the results showing here, you could see that the orange segment compared to, say, the purple segment really drove the expression of the reporter, indicating that it could be a potential enhancer. So now that we know that these small pieces of DNA have the ability to boost transcription of a gene, the question arises, how exactly do they do this? So how do enhancers communicate with their target promoters in order to regulate their transcription? As I mentioned before, was that enhancers can exist at varying distances from their targets. So they can operate when they're located proximally or very close to their target. And they can also function when they're very distant from their target. So how would a proximal enhancer versus a distal enhancer communicate with its target? Now, there are a couple of classical models of how an enhancer can communicate with its promoter. Two of these models are called tracking and linking. In the first model, scientists have this idea of how an enhancer can communicate with its promoter, where you have the binding of transcription factors to the enhancers. And then these enhancers track along the intervening DNA between the enhancer and the promoter region, recruiting various regulatory factors, including histone modifications, and this ultimately leads to the activation of the gene. Alternatively, with the linking hypothesis, it acts in a very much, they hypothesize that instead of transcription factors tracking along the DNA, perhaps they just oligomerize and form a bridge connecting the enhancer to the promoter, ultimately leading to the transcription of the target gene. So how would you test these different models of enhancer target communication? Also, does it matter what experimental approach you use? And also, does it matter what the system is? So one of the first questions, or one of the first ways that people started to look at the interactions between enhancers and targets was just by asking simply what binds to an enhancer. One experiment that can tell you this is something called a chromatin immunoprecipitation, or a CHIP. So here you have a cell where you have two regulatory regions, we call these enhancers, and they bind two different transcriptional regulators. So the way this assay is performed is, first, you chemically cross-link these proteins to the DNA with something called formaldehyde. Then you lyse open the cells and break the DNA into very small fragments, around 200 nucleotides. So what you get in the end is a series of fragments, some that don't have anything bound to them, and others that have these regulatory proteins cross-linked to them. So say you want to focus specifically on one of these regulators. So what you would do is introduce an antibody that recognizes one regulator versus another. So say you want to target regulator yellow, or A. So you introduce an antibody to regulator A in order to pull it out, and also the DNA that's associated with it. Next, you precipitate out that DNA using those antibodies. You pull it down, and then you reverse cross-link so you can remove the protein and just keep the DNA that was associated with that protein. Then you amplify your specific piece, and then you can map that back to the genome and figure out what pieces, what different DNA pieces that particular regular protein bound to. So you can sequence and map. So in the early 2000s, one group did this looking at different enhancer promoter associated factors, and also factors that were associated with the DNA in between the enhancer and the promoter. So through a series of chip experiments, they looked at factors over time, and they saw that different regulatory factors, including SNF, and also RNA polymerase. They also looked at histone acetylation over time, and they saw that progressively they tracked the acetylation and the accumulation of these proteins along the intervening DNA between the enhancer and the promoter. And they saw that over time, there was a progression of the signal in a unidirectional manner all the way to the promoter until a stable enhancer promoter complex was formed. So this was some of the first evidence of how an enhancer could potentially interact with the promoter based on the different proteins that bind to the sequences at the enhancer and promoter and in between. Another question, or a different way to ask about how an enhancer can regulate its target promoter, includes, you could ask, what regions in the genome does the enhancer itself actually interact with? So in a variation of a chip experiment, what you could do is see whether or not your enhancer A interacts with this yellow piece of DNA. So in a manner similar to a chip, you cross-link proteins that are associated, you lyse cells. Well, you don't cross-link proteins, sorry. You cross-link the DNA, and then you lyse cells so that you have free fragments of DNA, and you also have pieces of DNA that were in close proximity to each other, cross-linking to each other. Next, what you would do is digest the ends and add biotin, a biotin label here. And then you would ligate the sequences together so you can form kind of like this loop sequence. Then you break your sequence down into DNA fragments and you pull down what's stripped avidin beads. So you're fishing out only the sequences that have been labeled here. And next, what you would do is sequence the fragments in order to generate a contact map. And this contact map will tell you exactly what regions that your enhancer associated with in the genome. And you could theoretically form a map that looks like this, or you could infer from this map what the DNA would look like. So say it would look very loopy, like here. So going forward, how would you would like to know probably how do enhancers work in the context of a system? Why is it important that they work? And then how do we study them in different model organisms? So before I go on to the next topic, do we have any questions? Is everything clear up until now? Thumbs up. All right. So in this next section, I'll be focusing on enhancers in the context of the globin locus. So as you can remember from what I talked to you about before, we're this group that actually discovered this concept of enhancers. The green gene that they were looking at was actually the globin gene. So what is globin and why is it important? So globin is essential to blood function. So globin is a key component of hemoglobin molecules, where the oxygen you breathe from outside enters red blood cells and binds to these hemoglobin molecules. And this is what hemoglobin looks like. It is a heterotetramer where you have 2 globin chains and then 2 beta globin chains, alpha globin chains and 2 beta globin chains. The alpha globin chains remain a consistent part of hemoglobin, but the beta chains actually change over developmental time. But also deregulation of globin can lead to a variety of disorders, including thalassemia, sickle cell, anemia, and also it can infer genetic resistance to malaria. Here I'm showing you a schematic of the beta globin locus. And you can see that there are 5 different globin genes at this locus. And what's important to know is that each of these genes gets expressed at a different time in development. So early on, alpha globin will associate with either epsilon globin at the early embryonic stages or gamma globin throughout fetal development. And then as time progresses in adulthood, you get more and more expression of beta globin. A key regulator of this switch in which globin is expressed developmentally is this locus control region. So this locus control region, or LCR, controls the expression of these beta globin loci. So it's a good candidate to study enhancer association over time. And this is, so you can see that the LCR can either in early embryonic development to get fetal hemoglobin, it'll associate with the gamma globins. And then in adulthood, you can see that it alternately begins to associate with beta globins. In one of the journal club papers, the system they used to study the dynamics of LCR interaction with the globin locus over time are erythroblasts. So here they essentially mimic the developmental path of globin expression. You can see that over time you get more and more hemoglobin synthesis as these cells differentiate from more immature CD34 positive cells into mature red blood cells. So how would you track LCR enhancer communication with beta globin genes over time? So what the scientists did in the paper is they essentially borrowed ideas from natural systems. One of the first techniques that they used was CRISPR, which I know a lot of you have learned about, where you have a Cas9 protein that associates with a guide RNA that leads to the targeting of this protein to specific DNA loci, where you can get cleavage or you can insert a piece of DNA in order to get transgenic cells. Another concept that they borrowed from, or another tool that they used, was this interest in the paper was this estrogen receptor and estrogen interaction. So normally estrogen is present on the outside of the cell, and then on the inside you have the estrogen receptor. Once estrogen comes in, it associates with the estrogen receptor to form a complex, which then dimerizes. Upon dimerization, it can translocate into the nucleus and then turn on the transcription of target genes. So what makes this system so interesting is that it allows you to control the movement of a transcription factor, which in this case is the ER, into the nucleus. So they co-opted this system in the paper in order to control the movement of a transcription factor into the nucleus. So I've shown you how you can control the location of a transcription factor, but how do you detect when and where its target is transcribed? And before I move on, does anyone have any questions about the tools I've shown you so far? So that includes CHIP, the estrogen receptor, and then also CRISPR. Okay. So how do you know when and where a gene is transcribed? One of the ways to do this is to detect the transcripts with antisense probes in a process known as in situ hybridization, or ISH. So the way this works is you have an RNA probe that is antisense to your target mRNA, and this antisense probe is labeled with either digoxin or fluorescent dye. So what you do is that you introduce this probe into your cells, you wash, and then you use an antibody against your marker, against digoxin, and you have this antibody coupled to a marker. So the question is exactly what is the marker to be able to recognize this, to show where this antibody is. Turns out for this particular system that the marker is an enzyme. So this enzyme will process a substrate, which ultimately leads to the production of a product. Here. And what this looks like is what I'm showing you here with a Drosophila embryo. You can see here that this particular mRNA, which is Fushitarazu, you can see that this particular mRNA is expressed in this very stereotypical striped pattern in this Drosophila embryo. Any questions? Okay. So this leads me to the next topic, which is fly embryogenesis at this. Specifically, I'll focus on the snail locus. So in our second Journal Club paper, researchers probe the different enhancer dynamics in the Drosophila embryo. So in Drosophila, during early embryogenesis, it's characterized by a syncytium. So what this means is that you have nuclear divisions occurring, but you don't have the presence of cell membranes. So this is essentially a bag of nuclei that coordinate their activity by using different morphogens and transcription factors and enhancers. So as embryogenesis progresses, you begin to see the appearance of these different segmented regions. So segmentation is a sequential process that's directed by many genes that include various proteins, including transcription factors and different secreted molecules. So the pattern of expression that dictates the location of these segments is actually established very early on here when the embryo is still a syncytium. So these genes, they regulate, that are expressed here, regulate the segmented body plan of the embryo along the anterior-posterior axis. So the reason why these segments are so important are that they ultimately lead to the final anatomical segments of the fly. So you can see here that it matches pretty closely. So one way that scientists figured this out was using the same technique, the ISH, to detect the localization of these different genes that are expressed in the early embryo and then over time. And one thing that's very striking is that these genes are expressed in these very stereotypical stripes and have very specific locations in the embryo. So in one of the Journal Club papers, what they focus on is one of these factors called SNAIL. And you can probably guess why it's called SNAIL. So SNAIL is a highly conserved gene that encodes a transcription factor that predominantly functions to repress the expression of a number of genes in the ventral region of the embryo in order to promote the mesoderm fate. So here I'm showing you an embryo along the dorsal ventral axis, but if you were to take a cross section and then rotate that 90 degrees to look at you, what you would see is that SNAIL is expressed very strongly on the ventral region. And what's indicated here is that this portion of the embryo actually turns into the mesoderm. So here's an in situ of this view, and you can see that as the embryo develops progressively, that the mesoderm begins to form here, and you still have SNAIL expression here. One of the key functions of SNAIL, the key to the function of SNAIL is actually due to these zinc fingers in its structure that allow sequence-specific binding. So what's cool about these zinc fingers is that researchers have actually co-opted this in order to utilize them as genome editing tools. So they've used them to induce targeted cutting of loci, and also to target specific proteins to specific loci. So you can think about zinc fingers as a precursor to CRISPR. So now that we know that SNAIL regulates its targets, but also how SNAIL itself regulates, but what we would like to know next is how SNAIL itself is regulated. So here I'm showing you, once again, an in situ of snail in the Drosophila embryo, and you can see that it has a very stereotypical expression pattern. So this is the same embryo, just slightly rotated. So as you could have guessed, the expression of snail is regulated by enhancers. So like I mentioned before, the way these enhancers were characterized were very much like how I described earlier with the enhancer reporter assay. So here I'm showing you the a reporter assay where the LAC-Z gene is upstream of one of the presumed enhancers of snail, this region that's proximal to the gene here in orange. And you can see that the reporter is expressed in very much the same region that snail is expressed. And when you do the same with this distal portion of the genome here in pink, you can see that it also drives a very LAC-Z expression in a very similar pattern as snail. So just because you have these two snippets of DNA being able to drive the expression of a reporter in the same pattern of snail doesn't mean that they necessarily regulate snail. So another way that these researchers looked at this was to see what happens when you delete these regions. So here, once again, I'm showing you in blue, I'm in an in situ of snail, and down here you have snail represented in green, and then a marker of a gastrulation gene in red. So you can see that, you can imagine the embryo kind of closing in and on itself and creating the digestive system. So when you delete either the proximal or the distal enhancer here, what you see is a defect in the expression, the localization of snail. So you can, if you delete the proximal promoter here, you can see kind of a widening of the region in which snail is expressed, and it's not as precise as you would see normally. But when you delete the distal enhancer, which is here, you see that snail expression is severely disrupted, and this gastrulation marker is also deregulated as well. So the difference between these two enhancers is that one is considered the primary enhancer of snail, as it's proximal, and this other enhancer, this peak enhancer, which is distal to snail and located in a separate gene, is actually considered a shadow enhancer. So a shadow enhancer is essentially a redundant enhancer, and they tend to be remote secondary enhancers compared to proximal enhancers. And it's thought that they ensure the robustness and they make the expression pattern of their target genes more reliable, and they kind of decrease the variability of that expression. And so they are very important in development. So here I've shown you deletion experiments to look at the function of these two different enhancers. But another tool that is utilized in this journal, in the journal club paper that we will discuss, is something called an insulator. And it's specifically this gypsy insulator. And it's used as a tool essentially to, instead of deleting the enhancer, you can block its activity on its target promoter by introducing the enhancer. So this is something to look out for in those journal club papers. Do we have any questions up until this point? I've covered a number of topics, including not only enhancers, but the globin locus, but also the Snell locus in fly embryogenesis. Is everyone following? Okay. So now onto the last topic, which is transcriptional bursting. And so showing you this picture again, this is the progression of early Drosophila embryogenesis. As I mentioned before, this is a syncytium where there are no cell boundaries, and these are all nuclei that are actively dividing. So one thing that they really emphasize in the paper is that they look at this particular stage where all the nuclei essentially form a monolayer, and they are synchronized in the cell cycle. So this is actually probably the best time to do live imaging. And they did live imaging, and one of the things that you can see is that during this time in development, the embryos have a very synchronized way of expressing, but yet you can see these bursts of transcription occurring as indicated by these green dots. So what exactly is a transcriptional burst? There are several different features of a transcriptional burst, which include the duration of the burst, which is the rising period that corresponds to the time when the transcripts are synthesized. The mean intensity of the burst, so how many mRNA transcripts are occurring during that burst of transcription, and also how long the pauses are in between the burst. So there are two descriptors of burst, which include bursting size. So it's the magnitude of the burst. So essentially, this, if you were to measure the intensity of these green dots here, that indicates how many mRNA transcripts are actually being actively transcribed, and that would be indicative of bursting size. So that, along with how long that burst lasts, or that signal lasts over time, that collectively is considered the bursting size. Another feature that you should pay attention to when it comes to transcriptional bursting is how many bursts occur over a certain amount of time. So not only the bursting size, which is indicated here, but also how many bursts you get over the course of time. There are a couple of ways to observe transcriptional bursting. One that they utilize in the papers is single molecule fluorescence in situ hybridization, which is similar to what I discussed previously. The way they utilize this is to design probes specific to either the enhancer in one color and to the gene, which is in another color. And then you get binding of these probes to the enhancer and the gene. So you can see enhancers in red and say your gene in green. And after imaging, what you could imagine seeing is nuclei. Your enhancer would be labeled in red, your target gene in green, and you can look at their location relative to each other, whether they're next door or overlapping. Another way to look at transcriptional bursting is with this live imaging system known as MS2. So the way MS2 works is that you utilize this MS2 binding protein, which is tagged with GFP, and you express your RNA of interest where it has at its end MS2 hairpins. So imagine if you're interested in developmental gene A, you introduce MS2 hairpins to it, and you also, in the same cells that you transfect this plasmid with, you introduce this plasmid as well, so that this MS2 binding protein becomes transcribed and actively binds these MS2 hairpins. So there you have it. And you can see that this is a very clever way to do live imaging of active transcription in your cells. So you have your target mRNA with the MS2 hairpins, and you have your MS2 GFP binding protein. And the more transcription you get, the more GFP expression gets localized to those transcripts. And the way this is imaged is you take a series of snapshots over time, and then use a program in order to generate a dynamic movie. And this results in this beautiful image this beautiful video that I showed you earlier here, where you can detect, in this case, this is a fly embryo that has transgenic Eve, which is one of those segment genes that I described to you previously, and it's actively being transcribed during embryogenesis here, indicated by these green dots. So one of the ways to look at transcription to regulate how this bursting is occurring is to also stop the bursting. And one of the ways to stop the bursting or stop transcription is with actinomyosin D. That is essentially a drug that intercalates into DNA and prevents transcription from occurring. So this is another tool that they used in the journal club papers that we will be discussing. So in short, a couple of questions to keep in mind as you read the papers is, how do enhancers communicate with their target promoters? How does an enhancer regulate the transcriptional bursting of a target gene, and if it actually regulates the transcriptional bursting of a gene? And also, how do enhancers and genes interact to coordinate gene regulation in general? So with that, I'll take any questions. So does anybody need any clarification on the topics or the methods that I presented here? Because these are very important for understanding the journal club papers that we read tomorrow and Wednesday. Okay, everyone's good? All right, I see thumbs up.
All right, then we're going to take a break for lunch. And then this afternoon, you'll be working principally with Ahelia, who she did have her, I guess her video's off now, to actually start doing some of this yourself. There she is, so that you can see what it's like to actually look at data and begin to analyze it and trace chromosomes in nuclei. So I think that'll be fun. All right. You can always
put the Zoom link in the chat for you guys.
Ah, great. Okay. Good. All right, I'm going to sign out here. And Ahelia, I'm going to assume you don't need me this afternoon. I have more lectures to prepare, but if that's not the case, you should get it out.
No, it's fine.
Okay, good. All right, then I'm going to close out this meeting and say goodbye.